All right, if you're gearing up for your micro exams and the word poliomyelitis makes you just a little bit nervous, you are in exactly the right place. We're about to break down everything you actually need to know, from how the virus attacks to the global mission to wipe it off the face of the earth.
So here's our game plan. First, we're going to profile the enemy, the virus itself. Then we'll trace its destructive path through the body, learn how to spot the clinical signs, and see how we nail the diagnosis in the lab. And finally, we'll get into the legendary vaccines and the global endgame to finish this fight for good.
Okay, Section 1. Let's get right into it. Before we can really understand the disease, we have to understand what causes it. So let's meet our antagonist, the poliovirus. Okay, right off the bat, here is a massive counterintuitive fact you have got to lock in for your exams. The classic image we all have of polio, the paralysis, that is the absolute worst-case scenario. It actually happens in less than 1% of all infections. The other 99% of the time, this virus is a completely silent invader. The name of the disease basically tells you its entire strategy. Polio means gray matter, and myelitis means inflammation of the spinal cord. This virus specifically targets and destroys the motor neurons in the anterior horn. That's the command center for all muscle movement. When those neurons die, the connection is just severed. No signal means no movement. It is that brutally direct.
And when you look at it, it's a masterpiece of sinister simplicity. We're looking at a single strand of RNA, its genetic blueprint, packed inside this protein shell, or capsid. And that shell is just made of 4 key proteins. That's it. It's no fancy envelope, it's just a tough, simple, incredibly effective design. So if you're making flashcards, these are the absolute non-negotiables. It's a Picornavirius, which literally means pico for small and RNA for its genome. Its non-envelope structure makes it incredibly tough. And remember that VP1 protein? Well, that's the key identifier that splits the virus into 3 distinct serotypes. And this part is critical. Beating one serotype does absolutely nothing to protect you from the other 2.
Okay, so we know what the virus is. Now, how does this tiny little invader manage to launch a full-scale assault on the central nervous system? Let's trace its path, step by step, from entry to paralysis.
The entry is almost always through the fecal-oral route, so think contaminated food or water. Now, respiratory spread is possible, but it's way less common. For any exam question you get, your go-to answer for transmission has to be fecal-oral.
This sequence right here is pure gold for your exams, so let's walk through it. First, the virus gets into the gut and sets up shop in lymphoid tissues like the Peyer's patches. Then it uses this specific key, the CD155 receptor, to unlock and get inside our cells. From there, it breaks into the bloodstream, that's what we call viremia, and hitches a ride to the central nervous system. And that's where it finds its primary target, the anterior horn motor neurons. The virus hijacks the neuron, shuts it down, and the neuron dies. The result? Irreversible flaccid paralysis.
So that's what's happening on the microscopic level. But what does a polio infection actually look like in a patient? Well, the answer is this huge spectrum of outcomes, where that paralysis we all fear is really just the tip of a very, very large iceberg. This chart tells such a crucial story. Look at that huge blue slice. Over 90% of the time, the virus just wins, silently. It infects, it replicates, it spreads, all without the person feeling a single thing. And that's what makes it so difficult to track. A tiny fraction get a minor flu-like illness. And then you see that tiny, tiny sliver, less than 1%, who develop the devastating paralytic disease polio is so famous for.
And for that unlucky 1%, this is one of the classic clinical signs you need to look for, the tripod sign. The paralysis is characteristically asymmetric and descending, hitting the bigger proximal muscles first. So because the trunk muscles are too weak to hold them up, the patient has to plant their arms behind them just for support, which forms a tripod. It is a textbook sign of motor neuron destruction.
Okay, so you see a patient with acute flaccid paralysis. You suspect polio. How do you actually prove it in the lab?
So to confirm the diagnosis, you've got to find the virus. And the best place to look is a stool sample, because the virus is shed there for weeks. The absolute gold standard is to take that sample and introduce it to a culture of living cells, usually primary monkey kidney cells, and just watch. If the virus is there, it'll kill the cells, creating visible damage that we call cytopathogenic effects. A much faster alternative these days is to use PCR to directly find the virus's RNA. And that brings us to what is maybe the most important and definitely the most tested on part of the whole polio story, the vaccines that changed absolutely everything.
You absolutely have to know this table. On one side, you've got SOX-IPV, an inactivated or killed virus given by injection. A good way to think of it is safe but selfish. It makes antibodies in the blood, so it protects the person who gets it, but it doesn't stop the virus from replicating in their gut, so no herd immunity. On the other side is Sabin's OPV, an oral, live attenuated vaccine. Its genius is that it mimics a natural infection. It creates powerful IgA immunity right there in the gut, and it can even spread to unvaccinated people, creating amazing herd immunity. But that power comes with a tiny, tiny risk. The weakened virus can, on very rare occasions, revert back and cause paralysis.
So, you need a quick way to remember which is which when you're under exam pressure? Try the opposite trick. Sabaleek has al in it, which sounds like oral, but it's the opposite. It's the injectable one. Saban has an I in it, like injectable, but it's the opposite. It's the oral one. It's a simple trick, but hey, it could save you a mark.
Thanks to these incredible vaccines, humanity is on the verge of doing something truly extraordinary, eradicating a human disease for only the second time in history, right after smallpox.
And the progress has been just breathtaking. This is a true public health triumph. We knocked out wild poliovirus type 2 back in 1999. A massive milestone came in 2014 when India, once the world's epicenter, was declared polio-free. Then, in 2019, wild poliovirus type 3 was gone too. We have literally cornered the enemy, with only one of the 3 original wild strains left in the world.
So the final frontier for this wild virus is now just 2 countries, Pakistan and Afghanistan. For years, the acronym to remember was PAN, Pakistan, Afghanistan, and Nigeria. But thanks to incredible efforts, Nigeria reported its last wild case way back in 2016. So this leaves just 2 nations, where the final push is happening under some of the most challenging circumstances you can possibly imagine.
And this leaves us with a final, really thought-provoking question. You see, the endgame isn't just about hunting down the last case of the wild virus. The very tool that got us this far, the live oral vaccine, can, on rare occasions, circulate and regain its power, causing outbreaks of what we call vaccine-derived poliovirus. So the ultimate challenge is this: how do we eliminate the wild virus, while at the same time planning for a world where we have to stop using the live vaccine that helped us win? That's the complex final move in this global chess match against polio.